Hello friends, today in this video we are going to discuss about the MCQs on immunology, and this is the 12th part of the series of more than 1800 MCQs on immunology. Okay, so and this will be beneficial for many life science exams. Okay, so let's start the MCQ session on immunology. So the first question is the following is a primary mediator of anaphylaxis. The options are SRSA, SARS-A or serotonin, leukotrienes or prostaglandins. The right answer is option B, that is serotonin. So the following, that means the serotonin is the primary mediator of anaphylaxis. So what is anaphylaxis? Anaphylaxis is a life-threatening condition closely linked to IgG antibody activation of mast cell with subsequent release of preformed mediators including histamine, neutral proteases and proteoglycans from intracellular granules. And histamine and serotonin is thought to be the primary mediator of anaphylactic shock. Okay, so what is the mechanism of anaphylaxis? So IgE molecule, IgE antibody first bind to the surface receptor of mast cell and basophils, and then antigen combine with cell bound to IgE like this. This is the antigen bind to the IgE molecule which is already bound to the mast cell. Okay, and in next cross-linking between adjacent antibody molecules and then increase permeability of cells to calcium ions, and at the end the degranulation done and release of inflammatory mediators like this. That means inflammatory means histamine or serotonins. Okay, so this is the all about anaphylaxis. Okay, so move on to the next question, that is monoclonal antibodies are produced in hat medium, MEM medium or HE agar or CLED agar, and the right answer is option A, that is hat medium. So monoclonal antibodies are produced in hat medium. So what is monoclonal antibodies? So monoclonal antibodies are the antibodies that are made by identical immune cells that are all clone of a unique parent cells. Okay, and monoclonal antibodies can have monovalent affinity in that they bind to the same epitope. So these monoclonal antibodies have monovalent affinity, that means they only can bind with the only one type of epitope. Okay, and they are produced by unique parent cell. Okay, so they are monoclonal. And how we can produce monoclonal antibodies? So at first we have to infect a mouse with a antigen, and this mouse raise the antibody against that antigen, and then we collect those antibodies which are produced by this mouse, and then we fuse those antibodies with the myeloma cells and produce the hybridomas, because these myeloma cells can continuously produce those antibodies. Okay, so that's why we fusion with the antibodies with this myeloma cell and produce hybridomas, and then these hybridomas are cultured in hat medium to select for positive cells, and then we get the harvest the monoclonal antibodies like this. Okay, so this is the procedure of producing monoclonal antibodies. So move on to the next question, that is hat medium contains hypoxanthine, alanine or transaminase, or hypoxanthine, aminopterin, thymidine, or hypoxanthine, aminopterin or trypticase, or hypoxanthine, amine or telluride, and the right answer is option B, that is hat medium contain hypoxanthine, aminopterin and thymidine. So this hat medium, that is the hypoxanthine, aminopterin, thymidine medium, is a selection medium for mammalian cell culture which relies on the combination of aminopterin, which is a drug that acts as a powerful folate metabolism inhibitor by inhibiting dihydrofolate reductase. Okay, so it is a aminopterin is a folate inhibitor, and this hypoxanthine is a purine derivative, and thymidine is a deoxynucleoside. Okay, so move on to the next question, that is C2 and C4, which are the complement protein are synthesized in liver, spleen, macrophages or intestinal epithelium? And the right answer is option C, that is macrophages. So C2 and C4 complement proteins are synthesized in macrophages. Next is C3, C6 and C9 are synthesized in liver, spleen, macrophages or intestinal epithelium. The right answer is option A, that is in liver. Okay. So next one is the following is a zoonotic disease, that is plague, diphtheria, meningitis or poliomyelitis, and the right one is option A, that is plague. So plague is a zoonotic disease. So what is zoonotic disease? Zoonotic disease means also called zoonosis, and any of a group of disease that can be transmitted to humans by non-human vertebrate animals such as mammals, birds, reptiles, amphibians and fish. So zoonotic disease transferred from the animal to the human. Okay, so move on to the next question, that is passive immunization is indicated in tuberculosis, enteric fever, diphtheria or all of the above, and the right answer is option C, that is diphtheria. So passive immunization is indicated in diphtheria. So passive immunization or passive immunotherapy is a process in which individuals receive antibodies from another source rather than producing those antibodies on their own. Okay, that is the passive immunization. So next question is clinical infection leads to active natural immunity, active artificial immunity, passive natural immunity or passive artificial immunity? The right one is option A, that is active natural immunity. So clinical infection leads to active natural immunity. So here we can see acquired immunity are two types, active immunity and passive immunity, and in passive immunity that is also two types, that is natural active natural immunity or active artificial immunity. Active natural immunity we can get from antibodies developed in response to an infection. So that is the active natural immunity. So option 1 is the correct one. Okay, so move on to the next, that is most efficient complement fixing antibody via the classical pathway is IgG, IgE, IgM or IgA, and the right one is option C, that is IgM is the most efficient complement fixing antibody for classical pathway of complement. Okay, so move on to the next, that is J chain is present in IgA, IgM or both of the above or none of the above, and the right answer is option C, that is both of the above. So J chain is present in IgA and IgM. So here you can see in IgM here the J chain is present and IgA here the J chain is present. Okay, so move on to the next, that is factor B, factor D and properdin are components of classical complement pathway, alternative complement pathway, both of the above, none of the above. The right answer is option B, alternative complement pathway. So factor B, factor D and properdin are components of alternative complement pathway. So here this is the alternate complement pathway. Here the pathogens are directly bound to the C3b protein, and then C3b protein is activated by factor B, factor D and properdin, and then it converted to the C3b uh, converted to the C5 convertase, and then it bound to the C6, C7, C8, C9 and it produced the membrane attack complex and lyse the cell. Okay, so this is the way of alternative pathway. So move on to the next question, that is the following immunoglobulins is present in atopy, that is IgG, IgM, IgA or IgE, and the right one is option D, IgE. So the antibody or immunoglobulin present in atopy is IgE. So what is atopy? Atopy is a genetic disposition to develop an allergic reaction such as allergic rhinitis or asthma and produce elevated level of IgG upon exposure to an environmental antigen. Okay, so this is the atopy, that is the allergic reaction. And move on to the next, that is secondary immune response has a negative phase. Antibodies are mainly of IgG class or has a rapid or sudden rise in antibody titer or all of the above. So secondary immune response, option is D, all of the above. Okay, so for secondary immune response, suppose in the first when our body get exposure to an antigen, get exposure to a antigen first time, that time the response is like this. And in next time, second time, when the same antigen get the exposure to our body, then that time it gives quicker and faster and more rapid and more greater or larger response in secondary exposure. Okay, so this is the secondary immune response. So move on to the next, that is B lymphocyte. B lymphocyte form SRBC rosette, have thymus specific antigen on surface, undergo blast transformation with endotoxin, or are all of the above, and the right answer is option C. So antigenetically stimulated B lymphocytes undergo blast transformation to become plasmablast, and they are intermediate short-lived transitional cells which later become plasma cells. Plasma cells are non-proliferating and long-lived cells that secrete antibodies. Okay, so move on to the next one, that is the following is type 2 hypersensitivity. The options are atopy, Arthur's reaction, cytotoxic, or serum sickness, and the right one is option C, cytotoxic. So the following, that means types 2 hypersensitivity is cytotoxic, and this type 2 hypersensitivity involves IgG or IgM included damage to self cell. Okay, and either IgG or IgM is made against normal self antigen or a foreign antigen resembling some molecule on the surface of the host cell. Okay, so next one is when antibody and a particulate antigen combine, the reaction is precipitation, agglutination, adsorption, or neutralization, and the right one is option B. So agglutination. So when antibody and a particular antigen combine, the reaction is agglutination. Okay, so what is the agglutination and precipitation? So they're similar in they both act act as a antibody and antigen complex. So they both produce an antigen antibody complex, but the difference is nature of antigen. In precipitation, the antigen is soluble antigen, and in agglutination, the antigen which is present, that is insoluble antigen. Okay, so this is the main difference. So move on to the next, that is endotoxin is heat labile, is protein, polysaccharide, lipid in nature, or has specific tissue affinities, or has enzymatic action, and the right answer is option B, that is endotoxin is a protein, polysaccharide, lipid in nature. Okay, so endotoxin basically present in gram negative outer membrane of the gram negative organisms, here like this. These are the endotoxin. So when the cell is dead, that time the endotoxin get into the environment. Okay, so this is the endotoxin. So next one is immediate hypersensitivity is anaphylaxis, cytolytic, or immune complex disease, or all of the above, and the right one is option D, all of the above. So immediate hypersensitivity is anaphylaxis, cytolytic, immune complex disease. Okay, so next one is immediate hypersensitivity appears and recedes rapidly, or desensitization is difficult, is induced by mitogens, or cannot be passively transferred by serum, and the right one is option A. So it appears and reduces rapidly. Okay, next is immune reaction is triggered by immunoglobulins, complement, antigens, or lymphokines, and the right one is option C, antigens. So immune reaction is triggered by antigens. Next one is the following is a live vaccine: TAB, BCG, Salk, or Sample. So the right answer is option B, BCG. So live vaccine uses a weakened or attenuated form of the germ that causes a disease. So that is a live vaccine. And BCG, that is the Bacillus calmette guerin, is a prepared form of strain of the attenuated, that means the virulence reduced, live bovine tuberculosis bacillus mycobacterium bovis that has lost its ability to cause disease in humans. Okay, so this is the BCG vaccine, which is a live vaccine. So move on to the next, that is macrophages are activated by CD4 plus T cells or activated by lipopolysaccharide or can exhibit MHC class 2 antigens or are all of the above, and the right one is option D, that is macrophages are all of the above. That means macrophages are activated by CD4 plus T cells, are activated by lipopolysaccharides, can exhibit MHC class 2 antigens. So macrophages are a type of white blood cells of the innate immune system that engulfs and digests pathogens such as cancer cells, microbes, cellular debris, and foreign substances. Okay. So next question is, Thymus confers immunocompetence to lymphocytes, is responsible for humoral immunity, secretes hormone thyroxine, or are all of the above? And the right answer is option A. That is, Thymus confers immunocompetence to lymphocytes. So what is immunocompetence? Immunocompetence is the ability of a body to produce a normal immune response following exposure to an antigen. So here we can see the thymus located in the chest cavity behind the breastbone. Here the thymus is present and it produces thymosin, stimulate the development of T cell. Okay. So next question is, type 2 hypersensitivity is involved with hemolytic anemia, agranulocytosis, both of the above, none of the above. The right answer is option C, that is both of the above. So type 2 hypersensitivity is involved with hemolytic anemia and agranulocytosis. So agranulocytosis happens when your body doesn't make enough white blood cells called neutrophils, and these white blood cells fight with germs that make us sick. Okay. So the last question is, ELISA requires micro quantities of reagents, a substrate, elaborate washing step, all of the above. And the right one is option D, that is all of the above. So ELISA required all of this. So what is ELISA? ELISA is the basic assay technique known as enzyme linked immunosorbent assay, also referred to as EIA, that is enzyme immunoassay, that is carried out to detect and measure antibodies, hormones, peptides, and proteins in the blood. So here what is the main procedure? So at first this is the antigen, which is our main interest. So we detect this by adding a antibody, particular antibody against it, and then uh, by a substrate, and it is attached with this antibody is attached with a substrate, with a component, and then when we add the substrate with this enzyme, it convert into a coloring product. So we can say that this antigen antibody complex is formed. So this is a process of detecting antigens or hormones or peptides or any kind of proteins in the blood. Okay, so this is the process. So thank you for watching this video, and we will see you on our next video, that is the part 13 of immunology MCQs. Okay, so thank you for watching this video.